So plastics pollution is a really big issue in our society. There is a lot of plastic pollution in our oceans and just in landfills in general. And not a lot of it is biodegradable. So if we have some product that can be degraded naturally, it could save us a lot of pollution that exists just in landfills, in the water, anything like that. So I'm working with a specific metabolic pathway called the ethyl malonyl-CoA pathway. And what's really interesting about this specific pathway is that it produces a bioplastics molecule. So we're trying to figure out when cells know how to turn this pathway on or turn it off, because if we can understand that, we could eventually manipulate it and then just sort of overproduce bioplastics to reduce plastic pollution. In my particular organism, we're only looking at the one pathway, but there are other organisms that also have this pathway and also have other pathways of interest that essentially do the same thing, just don't produce the bioplastics organism. So we can then look at when do they use those pathways versus the bioplastics pathway, and how are they turning that off and on, and is it the same way? Is it a different way? Could we manipulate that one too? So that's one of our other projects that actually Erin's working on.
The project that I'm working on in particular is with the ethyl malonyl-CoA pathway, so the same pathway that Stephanie is looking at. And I am trying to identify when the bacterium that I'm looking at, when they use that process during their growth stages. And this pathway is the one that leads to bioplastic production. So if we can identify when the bacterium uses it, then we know when to target the overproduction of the organism. And it helps us tailor a potential manipulation for later on. It's very nice to have other people in the lab that are working on similar things. We are using the same pathway but investigating two different parts of it. But that part's also interesting because when I'm doing my project, I end up with questions, and some of those questions are actually what Stephanie's trying to answer. So it is interesting because it helps fill in a lot of the background pieces that you wouldn't necessarily have if it was just one person looking at one specific thing.
The biggest reward working with students is not the aha moment. While those are really rewarding, it's the synthesis of the aha moments. It's when they achieve that aha moment so regularly that they start to produce new knowledge. They start to see the world in a way that nobody else does. Even if it's ever so minute, if it's, just minusculely, they see it in a way that nobody else does because they understand something more than anybody else. That is the biggest reward. All of the work that we do here can end up with citizens who are better able to synthesize aha moments, see the world slightly better than the rest of us, and then improve it so that we can all understand how much better it can be.